You know that feeling, like we're always in a fight against getting sick? Well, it turns out we totally are, just on a microscopic level. Today, we're diving deep into the world of bacterial genetics. We're going to find out exactly how those superbugs, the ones that laugh at our best medicines, are actually made.
So, have you ever wondered about that? How do bacteria, these tiny little things, manage to outsmart our best medicines? I mean, it's a huge question, one that even top scientists are grappling with. How do they keep winning against our most advanced drugs? Let's get into it. Well, the secret is all in their DNA. And, maybe more importantly, in how they share it. It's kind of like they have their own secret internet. They can just pass around survival tips and instantly download the latest hacks for beating whatever drugs we throw at them.
Okay, so to really get this, we've got to look at what a bacterium is working with. Its genetic toolkit. The best way to think about it is like a smartphone. You've got your basic operating system, right? And then you've got all the extra apps you can download.
So, on one hand, you have the chromosome. That's the operating system. It's this one big circle of DNA that has all the essential, housekeeping genes, everything the bacterium needs just to live. But then, you've got the plasmids. These are the bonus apps. They're these little extra circles of DNA, and while they're not essential for day-to-day survival, man, they can carry some serious special abilities.
And what kind of bonus features are we talking about here? This is where it gets pretty wild. A plasmid can give a bacterium the code for antibiotic resistance, or the ability to produce toxins like the one that causes tetanus. It can even help them stick to our cells better or resist toxic metals. These aren't just minor upgrades. These are the features that turn a regular bacterium into a full-blown superbug.
All right, so bacteria can get new powers by grabbing these plasmid apps. But that's not the only way they change. They also have a way to evolve from the inside out, and they do it on fast forward.
And that happens through something called mutation. You can just think of it as a little change, a typo, in their genetic code. Now, sometimes these just happen spontaneously, totally random. But they can also happen when the bacterium is under stress. And what's one of the biggest stressors we put on bacteria? Yep, antibiotics.
And don't underestimate these little typos. A single tiny mutation can have massive results. Suddenly, that bacteria might be able to produce a new toxin or get way better at forming those protective biofilms. And of course, the big one, it could become resistant to an antibiotic.
Okay, so they can get new apps and they can mutate internally. But here's the part that is a total game changer. Bacteria don't keep their secrets to themselves. They share them. They have these amazing ways to pass their genetic tricks on to their neighbors. This is called horizontal gene transfer, and there are 3 main ways they pull it off.
So first up, we've got what you could call the bacterial handshake. It's called conjugation, and it's a way for 2 bacteria to directly connect and swap some DNA.
And you can see how it works right here. You have one bacterium, the donor, that has one of these special plasmids. It literally reaches out and builds a little bridge to a recipient cell and then sends a copy of that plasmid, one of those bonus apps, right across.
Let's walk through it step by step. First, that donor cell grows this little tube. It's called a sexpilus to connect to the recipient. That forms the bridge. Then, a single strand of the plasmid DNA gets transferred over. Once it's inside the new cell, the recipient just synthesizes the other half. And boom, it's got a complete copy. And what's really crazy is that now this recipient cell often has the ability to become a donor too and pass that trick on to the next bacterium.
Okay, so that was the handshake. Next up is transformation. And if conjugation is a handshake, this is more like scavenging for treasure. It's all about bacteria just grabbing loose bits of DNA from the world around them. So picture this. A bacterium dies and its cell wall breaks down. All of its DNA just spills out into the environment. Well, another bacterium that's just hanging around can actually grab those free-floating DNA fragments and pull them inside, weaving them into its own chromosome. And just like that, it might pick up a brand new trick.
Now, not every bacterium can do this all the time. To pull this off, they need to be in a special state called competence. Think of it like they have to turn on their DNA vacuum cleaner. Some bacteria are just naturally competent, but for others, it's a switch they only flip when things get tough, like when they're stressed out.
All right, our third and final method is called transduction. And this one is wild because it's not just about bacteria to bacteria. This one involves a middleman, a virus, that acts like a delivery service. And this delivery vehicle is a very specific kind of virus called a bacteriophage, or just phage for short. Now, phages are basically tiny little robots programmed to do one thing: infect bacteria.
But sometimes they make a mistake during their assembly process, and that little error can end up being a huge win for a different bacterium down the line.
Here's how it goes down. A phage latches onto a bacterium and injects its own DNA. It hijacks the cell to make tons of new phages, but during assembly, whoops, a mistake happens. One of the new phages accidentally gets packed with a piece of the bacterium's DNA instead of the virus's DNA. Then, the cell bursts open, releasing all the new phages, including that one faulty one. So when that faulty phage goes and infects a new bacterium, it's not delivering a viral payload. It's delivering genes from the last bacterium it was in. It's basically an accidental gene delivery service, all run by a virus.
So, okay, we've got conjugation, transformation, transduction. These are all incredible ways bacteria evolve and share information. But let's bring it back to the real world. Why does any of this actually matter to us?
This right here, this is why it matters. Let's connect the dots. That plasmid transfer we talked about, that's how antibiotic resistance spreads so fast. It's a huge problem. Those little mutations, that's how new, maybe even more powerful toxins can suddenly appear. And transduction and transformation, that's how bacteria can pick up new dangerous traits, or how pathogens can suddenly learn to resist our drugs. Every single one of these mechanisms is a direct challenge to human health.
So the big takeaway here is that bacteria are not just sitting around waiting for things to happen. They are constantly learning, evolving, and sharing survival strategies. It's happening all the time at a speed that is just alarmingly fast, way faster than we can invent new medicines. And this really gets to the heart of the problem. As Dr. Ndifria Kabasi Robinson says, horizontal gene transfer is a major way bacteria acquire antibiotic resistance genes. It's not just a side story, it is the main way. This sharing is the engine that is literally building the superbugs we're so worried about.
And that leaves us with a pretty heavy question, right? Bacteria are trading their survival secrets constantly. So in this biological arms race against an enemy that adapts this quickly, how are we going to adapt?